everyone. Welcome to the online lecture again. Tonight we'll talk about cell division. So we will briefly explain why cell will divide, the cell cycle. The cell cycle is the major part of today's lecture, so pay attention to that. And then we will talk about chromosome at the end, mitosis and meiosis. So these two are the major part. The cell cycle is important, and then the steps in mitosis and the step in meiosis, they are the most important part. So the introduction. During the Beijing Olympics, people discovered this. It is an algae that grow in the around the Beijing area, because Beijing is heavily polluted. So the water has a lot of chemical or nutrient for the algae to grow. That's why they have this. People see this and put this picture. And then the question is, how come those algae can grow so fast, and how come they can? In what method do they grow so quickly? So remember in the first lecture we talked about the 7 characteristics of living organism. One of them is that we are able to reproduce, and we are able to reproduce, and most importantly, we are able to grow. And I mentioned before there are 2 ways for us to grow. One is to increase the number of cells. The other one is increase the size of the cell. So tonight we will talk about how we increase the number of cells. So after, it takes a trillion of cell division to turn a fertilizer into an adult human. This is basically describing mitosis. And cell division is a generation of daughter cells from parental cells. Usually for mitosis, the parental cell and daughter cell, they are identical. But for meiosis, meiosis is the process of making we call it germ cells or the sex cells, like sperm and eggs. And usually the parental cells and the daughter cells, they are different. So we see this example on our skin. If you get too much sunburn, then you have the superficial layer of the cell. They will become dead. And if you take anatomy, you will learn in a much detailed level about the skin structure. We have a layer of cell that will continue to divide, and then it will replace the dead skin on the surface. So why cell divide? Well, cell divide for many reasons. So other than growth that I mentioned before, we can use cell division for asexual reproduction. For example, in prokaryotic cell, like bacteria and archaea, they use something called binary fission. Binary fission is basically very similar to mitosis. They use that for asexual reproduction. So the parent and the so-called the children or the daughter cell, they are identical. Mitotic division, well, for eukaryotic cell like us, we also use mitotic division. But for single cell eukaryote, like let's say amoeba and paramecium, they also use mitotic division for asexual reproduction. But for multicellular eukaryote, just like us, we use it for growth, repair, replacement of the cell tissue, just like the previous slide I told you about sunburn, and then the skin can replace the dead surface skin cell. For meiosis, I mentioned that it is for sexual reproduction because we use meiosis to make the sex cells, namely the sperm and eggs. So as I mentioned before, asexual reproduction will result in offspring that are genetically identical to the parent. But sexual reproduction will produce offspring that are different from the parents. That's why when you look at yourself and also your children will look different from you, but they will look very similar. They will look similar, but not identical. And also, even among your siblings, you and your brother and sister, they will not look the same. We'll briefly explain why your children, they do not look identical to you, and also your siblings, how come you look different from your sibling. Just briefly explain that later. So let's go back to talk about the asexual reproduction in prokaryote. Now, asexual reproduction begins with the first step is the replication of the genetic material. Remember, we call this the nucleoid. Nucleoid is the region in the bacteria that, or in the prokaryote, that contains the DNA material. They do not have a nuclear membrane to enclose this material. And then the first step is replication of this material in the cell. And then the second step is called cytoplasmic division. Cytoplasmic division is basically the pinching of the cell so that eventually you have two separate cells, two identical cells, so that the daughter cell, they look identical to the parent cell. But keep in mind that this is a very interesting point. For prokaryotic cells, they have cell wall. So when they are doing a cytoplasmic division, they actually have the new cell wall here. And they also have this pinching. In plant cell, when plant cell undergoing a mitotic division, they will have this. They call it a cell plate. This cell plate is like the precursor of the cell wall. And for animal cell, they do not have cell wall, so the cell will pinch. We call it cleavage furrow. In other words, bacterial cell, when they divide, they exhibit the properties of both plant and animal cell, because it shows the cell plate and also the cleavage furrow. So it is, I personally think it's very, very interesting. We will talk about the cell division for plant cell and cell division for the animal cell later. And then you will see that plant cell, they only have the cell plate, but no cleavage furrow. But animal cell, they have cleavage furrow, but no cell plate. For eukaryotic cell, we also use mitosis. As I mentioned, that mitosis will give the daughter cells that are identical to the parent cell. And it is always let me see. So the new term here is this cytokinesis. Cyto means cell. So cytokinesis basically is a splitting of the cell into two. And the nucleus divide is called meiosis. But actually there's another term, it's called karyokinesis. Karyokinesis, meaning that it is the splitting of the nucleus. So it is always karyokinesis occur before the cytokinesis. So similar to the bacteria, remember, first step they replicate or split the DNA into two, and then second step they split the cell. So for the eukaryotic cell, we do the same thing. The first step, we split the nucleus into two, karyokinesis, and then we split the cell into two, cytokinesis. So eukaryotic cell follows similar pattern as the prokaryotic cell. And we call this mitosis. Now we give you an overview of meiosis. So meiosis is the process, is a specialized form of cell division that make gametes. Gametes basically means sex cells. Of course, we know that they are the sperm and eggs. And for meiosis in animal result into egg in female. For male, it forms sperm. And I just want to bring you up into attention that egg, the plural, we have to put an S, the sperm, that we do not put S. So both singular and plural, it has no S. So sperm can be plural or singular. Meiosis reduces the genetic information passed to donor cell by half. So half we call it haploid. So if it is N, we call it haploid. Through N, we call it diploid. So what does it mean? We will talk more about the haploid. We talk about haploid and diploid on Wednesday's lecture. Right. We talk about this on Wednesday lecture. So remember how, so the question I asked on Wednesday, how many chromosomes we have and how many pair of chromosomes we have. So remember, we have 23 pair of chromosomes and we have 46 chromosomes. So when we have 23 pair of chromosomes, we call it a diploid. When we have 23 chromosomes, we call it a haploid. In other words, in sperm and egg, they have only 23 chromosomes. So if you have an egg that has 23 chromosomes and a sperm that has 23 chromosomes, and they fertilize and fuse together, then you will have 46 chromosomes. When you have 46 chromosomes, that means you have 23 pair of chromosomes. Then you will have diploid. So this is the so-called reproductive cycle of human being. So male, the testis will produce a haploid sperm. Female, the ovary will produce a haploid egg. After fertilization occurs, the zygote zygote becomes a diploid. And then the zygote will undergo mitosis and to develop the fetus. And I forgot to say that testis and ovary, they will undergo meiosis to make sperm and egg. So as I mentioned before, well, egg is haploid, sperm is also haploid. when you combine the haploid egg and the haploid sperm into one, then you get a diploid zygote. So gamete is the sex cells. We have a new term, the somatic cell. Somatic cell, soma. Remember what soma means? In the cell structure and function lecture, we talk about cell body. Cell body has a different name. It's also called a soma. So somatic cell is basically, we call it non-sex cell in the body, or you can just call it body cell. As long as they are not gametes, as long as they are not sperm and eggs, we can call them somatic cells. So most of the somatic cells, they are diploid to end. But most of the gametes, they are haploid, one end. So we already talked about it when a haploid sperm fertilize the haploid eggs, then they will become a diploid zygote, and then the diploid zygote will start to divide or undergo meiosis to increase the number of cells so that the embryo can develop and grow. Other than cell division, meaning that increase the number of cells, they also undergo something called cell differentiation. Cell differentiation is a process that cells grow into different specialized form of the cell. For example, the cell in our brain, we call it neuron. Neuron, the brain cell, they are very different from, let's say, our skin cell here. And the cell in that form our bone, the bone cell, we call it osteocyte. They are also very different from our neuron. So we call it cell differentiation. It's like, an analogy is like different people, we have different occupation. Some people, they are, well, like me, teaching. Some people, they are cops. Some people, they are the firefighter. Some people, they are the taxi driver. So, different people have different jobs, occupations. Similarly, different cells, they also have different functions. That's why we need to have a cell differentiation. So, the second point is basically says that in zygote, before the cells start to have a differentiation occur, all the DNA, all the 23 pairs of chromosome or 46 chromosome that we find in zygote, we can find the same 46 chromosome in, let's say, our skin cell. And the same 46 chromosome, we can find them in our neuron. Or the same 46 chromosome, we will find them in our osteocyte. So the germline cell, basically these are the cells that we find in our testes and eggs because they produce the gametes. And then stem cells is a term that we hear most of the all the time nowadays. Stem cells, they are unspecialized cells that can grow, regenerate, or repair tissues. You can find stem cells, let's say, when it is in the zygote, or you can find, so the cell, you can call the cell the zygote. They are kind of like stem cells. But in the placenta, in the fetus, there are a lot of these kind of stem cells because when the fetus are growing, they require stem cells to differentiate into different specialized cells. That's why some people, they would harvest stem cells from the placenta instead of from the embryo directly. Because when you harvest the stem cell from the embryo, it is inhumane. So one thing we want to tell you the difference is that the difference between male and female in terms of the meiosis. So for female, meiosis begins when the female, when she is a fetus. That means if you are pregnant with your daughter will already produce all the eggs that she will have for the rest of her life when you are pregnant with your daughter. But the last step of the meiosis, to complete the meiosis, occur during start during her puberty. So the menstrual cycle starts during puberty. Then each month, one egg or one gamete, one egg will go through the last step of meiosis, and then you will either become fertilized or you will become the menses. And then for male, meiosis begins in puberty, so we do not make our sperm when we were an embryo or fetus. So and for male, we make sperm until old age. We don't usually how do we say this? That's why many old people, they can still have baby. But for women, usually they stop the last step of they stop producing eggs, or actually they stop the last steps of the meiosis around the age of 50. We call it menopause. So there's a limit for female about about reproduction, the time limit. But for male, we can reproduce until the old age. Yeah, it sounds very unfair, but there's nothing we can do about this. Yeah. Well, actually, there is something we can do about this. Nowadays, we can freeze our sperm and eggs, so that the reason is that if women they try to have a baby after the age of, I would say, 35 or so, then the chances of having the chances of the baby having the Down syndrome will become very, very high. So it is better for women to have baby before the age of 35. Otherwise, the risk of having Down syndrome baby will increase dramatically after the age of 35. That's why many women, especially women who have a career, they like to, nowadays they would freeze their eggs when they were young, so that the healthy eggs can be kept until after they are married and they want to have kids, then they have the young healthy eggs that are in the freezer that they can use for fertilization and pregnancy. Okay, cell cycle. The first important thing. So cell cycle can complete in 90 minutes in the beast, but in human being it is approximately 24 hours. So it is easy to memorize. For human it is approximately a day, 24 hours. So what does it mean? This is the definition of cell cycle. It's basically tells you that how the cell goes from non-dividing into mitosis or meiosis. So as I told you before, it first occurs the nuclear division, karyokinesis, and then the second step is the cytokinesis. Karyokinesis is basically splitting of the nucleus or splitting of the chromosome. After the nucleus, after the chromosome inside the nucleus they are split, then the cell will proceed into cytokinesis to divide the cytoplasm into two, so that we'll have two cells and two nucleus. So we have G1S, G2 and phase, and then we have this. Some textbook call it the M phase. M phase stands for mitosis. And then we have the G0 phase. We'll briefly go over each phase. So one thing that I want to mention is that G2S and G1 phase, all these, we call it interphase. Interphase, during interphase what will happen? So takes in nutrient, manufacture protein, the cell can grow, grow the size, grow bigger, and it can do its function, specialized function, and most importantly, during interphase, it is for the cell to prepare for the cell division. So G1 is G stands for grow, so the cell grow. S stands for synthesis. Synthesis, what does it make during this S stage? It makes DNA. So DNA replication or DNA synthesis is during S phase. And G phase is also the second growth phase. After G2 phase, then it goes to the M phase or the cell division. I want to bring you up to an attention that you see during G1 phase, you see the chromosome, there are only it's only one piece of chromosome. Then during S phase, you have two pieces of chromosome joined together. And then G2, and then after cell division, then the chromosome becomes a single piece. So it says that the size of the cell and the protein content increase in G1, G2 phase. Why? Because after the cell, during the mitosis or after mitosis, one cell will become two. That's why you need to have the cell size needs to grow to at least double of the normal cell size, and also the protein content. That means the enzyme or whatever machinery inside the cell, they also have to double the number, so that after the cell division, each cell should have the normal amount of protein. So as I told you that the S phase, S stands for synthesis. That means the DNA replication is in S phase. So this is interesting because I asked this question for my physiology class, and lots of people, they couldn't answer this question. Actually, we covered in Bio 3, but I'm surprised many physiology students, they do not remember. So my question to them is that before cell division, we have how many chromatid? And after cell division, how many chromatid do we have? So in here, it is very clear. We call this a chromatid. Chromatid. So before cell division, or before cell replication, we have one chromatid. But after the cell replication, we have two chromatid. So we have three terms that many students like to mess up. Chromosome, chromatids, and chromatin. So for chromatin is the DNA that we see all the time, let's say during interphase. Not during interphase, during the G0 phase. G0 phase, we'll talk about it in the next few slides. But G0 phase is basically when our cells are not doing anything. When a cell are not doing anything, when a cell is not in the interface, and it's not in, when a cell is not in the interface, when a cell is not in the mitosis, that means when our cells they are mature, performing its normal function, we call it G0 phase. Chromosome occur in the interphase. So what happens is that when the cell exits G0 phase, it will go to the interphase. When the cell goes to interphase, then what happens is that the chromatin will condense and pack and coil, and wrap up itself, and then it will form chromosome. So we call this chromosome. Chromosome is basically formed by threads of chromatin. And chromatin, they are basically you may call them arms, or you may call it so you may call this the arms of chromosome. And then there are terms that also confuses students also, centromere. So centromere is the part in the middle of the chromosome. The middle of the chromosome that connects, you may call it connects the two, the upper and the lower chromatids. And it is also the part that mitotic spindle will connect to. We will talk about mitotic spindle during the, when we describe about the mitosis. Centrosome. Centrosome is basically made up of centriole plus some kind of gelatinous protein. And centriole, they are the So we will see centrioles in action when we talk about mitosis. Basically, centriole, they will shoot out some spindle, and then the spindle will connect to the centromere on both sides. So when the centriole, they contract or retract those spindle, then you will pull these two chromatins apart. When the two chromatins is separated, then it will it means it is the karyokinesis, splitting of the chromosome or splitting of the nucleus. So remember, before replication we have one chromatid. After replication we have two chromatid. But after the after cytokinesis, and after the karyokinesis and cytokinesis, we have only one prototype. So G0 phase. So I talk about G0 phase here. So during G0 phase, they call it the safe haven of the cells. And during this stage, the cell doesn't do the mitosis or meiosis. That's why I said that it is against the progress to cancer. Well, what is cancer? Cancer is, as you know, it's like uncontrolled cell multiplication. When your cell continue to divide non-stop, then you will have cancer. As I said, that G0 phase is basically the cell they are in a state that they do not divide. Because of that, if they do not divide, then that means there will be no cancer. So it says that G0 phase can last for a few days to the lifetime. It gives you two examples. The liver cell, divide only once a year on average, but neuron, the cell in our brain, they do not divide at all. Most of the neuron, they stay in G0 phase for our lifetime, but liver cell, they divide once a year. So it said that for DNA replication, the gene, they're usually not active during the G0 phase. It's very logical because G0 phase, we don't need the gene. We don't need the gene for DNA replication because the cell is not dividing. So, as I mentioned that for the cell, if the cell divides uncontrollably, that means we cannot stop the cell dividing, they will become cancer. Because of that, during the cell cycle, we need to have something called checkpoints. That means we need to have some safety procedure. We need to have some checkpoint here to regulate, to control the cell division. Otherwise, the cell will keep on dividing, then we will all have cancer. So we'll talk about some genes that control the cell cycle. We have something called proto-oncogene, and we have tumor suppressor gene. As the name suggested, proto-oncogene, they are the gene that, proto meaning that it's a primitive or primary. Oncogene means cancer gene. So proto-oncogene is basically a gene that will trigger the growth of the cancer. And the tumor suppressor gene, as the name suggested, it will stop the cell division, or it will suppress the gene from mutation, or it will suppress the cell division. So one, you can find yin and yang again. One is like a gas pedal. The proto-oncogene, you will just tell the cell, okay, keep on dividing. The other tumor suppressor gene is like the brake of your car. You will tell the cell, okay, stop dividing. You have enough cell, then you should stop. And for the triggering, for the activation of the oncogene, you need to have growth factor. Growth factor, they are basically some kind of a cell signal. For example, hormone is a form of a growth factor. When we were in puberty, we have a lot of hormone. And these hormones help us to grow tall and big. And that's why a growth factor is the one that can potentially activate the oncogene. So for example, when the stimulating growth factor binds to the receptor on the cell membrane, then this receptor will activate a signaling pathway, so that the signaling pathway will activate the oncogene in the nucleus. When the oncogene in the nucleus is being activated, what does it do? You make protein that will stimulate the cell cycle, meaning that the protein will tell the cell, okay, you better start to divide now. But for the tumor suppressor gene, if we have inhibiting growth factor that binds to the receptor, cell surface receptor, a different signaling cascade will occur, and this signaling cascade will actually also bind into the, it will also go into the nucleus, and then it will, if this piece of tumor suppressor gene here that will bind to this tumor suppressor gene, and if this tumor suppressor gene is mutated, that means the protein that it makes is not effective to stop the cell division, then you also cause cancer. So either you activate the oncogene or your tumor suppressor gene is mutated, then you will have cancer. In either case, you will also have cancer. Apoptosis means you can kind of similar to apoptosis is very similar to a programmed cell death. So it's basically a process that a cell kills itself. Yeah.